thank you to the organizers of the meeting, to Tracy, to Lennox-Gastaut Syndrome Foundation for giving me a chance to share with you some of our work and some ideas. So I've changed the title of my talk a little bit and even Twittered with it even during the conference. So the talk is now how to predict who will develop LGS, health data science, biomarkers, and LGS. These are my funding and disclosures. And here's an outline. I'll talk a bit about health data science, and then I'll give a brief introduction to biomarkers. The core of my talk, I want to spend some time talking about some natural history work we did with Lennox-Gastaut here in New York City. The data I'll share has been published in abstract form, and we have a manuscript that's coming together, but I didn't want to wait. I wanted to share some of those findings with this group and to use that as a launching pad to discuss 3 opportunities for early intervention. Tracy, I'm reflecting on your advice to us that we should focus on the treatment of active LGS. And I think that there's a tension in this talk and some others between this urge that many of us in the pediatric epilepsy community have to try to stop LGS from happening at all, as well as the need to treat people who actively have the disease. And I think that tension will be here in this talk as well. So let me start by talking broadly about health data science and what it is. Health data science is an academic investigation thinking about how big clinical and administrative data sets can help us think about and learn about questions that are important to us, both in clinical care and clinical research. So when we think about what those data sets are, you can think about claims data sets. You know, every time you see a doctor, there's a bill that's created. Some of those claims have been curated into data sets like the pediatric hospital inpatient system. Clinical data sets, I think that's where a lot of our heart lies. Electronic health records data, as an example, ways to aggregate those electronic health record data, for example, through learning healthcare systems. And EEG data is data we collect regularly, as well as MRI. Through our work, and those data are there for the taking for research. PCORI, the Patient-Centered Outcome Research Institute, has funded several initiatives, including the Rare Epilepsy Network, to gather electronic health data from a variety of sources. The ICU has an ocean of data created moment to moment on patients who are admitted there. And then there are other idiosyncratic data sets. Registries is incredibly important to our field. Those are often exquisitely curated data sets where people have gone through the records and tried to create analyzable data sets. And even birth records can be helpful. These kinds of large data sets are valuable to us in at least 6 domains: active clinical care, just having access to the data to make decisions about our patients; surveillance and epidemiology, thinking about incidence, prevalence, comorbidities, and mortality; health services research, which is its own academic area of focus that is interested in the organization, financing, and delivery of healthcare; comparative effectiveness research, which is to say we have 2 different approaches to take care of a child with LGS, as an example. Which one is better and by how much? Quality improvement. We think we know what we're supposed to be doing, but we're not doing it. Why? And how can we get better? And finally, related to clinical trials, which we will hear more about from Dr. Dlugos, finding patients to enroll in clinical trials, health data science can help us with that. A few examples. The Pediatric Epilepsy Learning Healthcare System is one of 2 epilepsy learning healthcare systems that are active. The pediatric epilepsy one is I lead in conjunction with Dr. Patel, who is on this conference, Dr. Berg, who's in the conference, and also Dr. Jeff Buchalter and Dr. Renee Schellhaus. The idea there is you take the electronic health record data from all of your sites, bring it together into a data warehouse, a clinical data, an electronic data coordinating center. And I think about those data in sort sort of three buckets. There's the structured data, the demographics, insurance, visit history, et cetera. That's all there for the taking if you can just figure out how to get it out. There's the clinical data, which in many ways is the hardest. This is questions like, is the patient still seizing? What kind of epilepsy do they have? Why do they have epilepsy? These are important pieces of information we write in our notes, and we all do it differently. So figuring out how to get that standardized through routine care processes is core to a learning healthcare system. And then the fine phenotype data, the MRI, the EEG, the genetics, adherence data as an example. All of that data, we do know how to get it out. It's just very expensive. You have to hire an army of research assistants and coordinators to pull it out. These data can then fuel inquiry into those 6 ideas that I mentioned before. And then what makes a learning healthcare system is you take that data and feed it back and try to engage in these virtuous cycles where you're consistently learning from what you're doing and making changes and trying to do better and better and improve outcomes. One of the problems I mentioned is getting the clinical data in. So as an example of the learning healthcare system work, we designed a standard form for use in clinical care by clinicians. And I just snapped some screenshots from a test patient during the conference to show it to you. This is in our active EHR system and is also being implemented. We've got it in 6 or 7 sites around the country with another dozen or so working to get it built. It's a bunch of standard questions. You can indicate that the child has Lennox-Gastaut syndrome. You can indicate what the gene is. And all of this data gets standardized. If we all do it in the same way, then it's easy to, well, it's conceptually easy to extract it out of the EHR, and then we can learn from our shared experience in a national or even international way. A second example is registries. The Pediatric Epilepsy Research Consortium has been really, really good at this. They had an infantile spasms registry that Kelly Knupp led. We were able to do some comparative effectiveness work with that, which again showed that for infantile spasms, first-line therapy, ACTH or prednisolone, vigabatrin, and nothing else really works. As another example, Dr. Berg, who's in the conference, had this exquisitely curated registry of early onset epilepsy. We used that data set to understand what was the most effective treatment as first line, and found that for infants 1 month up to 1 year, that levetiracetam was much better than phenobarbital with an odds ratio of like 4. It was a large effect size. And then as a third example, we're all sitting on EEG data. We all have smart grad students and motivated residents and fellows who spend time with us. And so doing EEG analysis is a tool that I think many of us have started to develop when we find someone who's talented and is able to do some of that work. So this is just an example. Peter Yan was a really bright adult neurology resident who rotated with us, and he used some median power spectrograph techniques to do some seizure identification, and then even used some machine learning techniques to try to automate the detection of seizures. That's the first foundation for what I want to talk about, that there's this ocean of data in our electronic health record systems for us to use. The second thing I want to talk about is biomarkers. A biomarker is a well-defined biologic characteristic that indicates normal or pathologic processes that changes with therapy. They don't have to be so fancy, or they don't have to be there's lots of examples of biomarkers. I wanted just to show one in pediatric epilepsy. That's something that the clinicians in the audience use regularly. This is a child who, when he fell asleep, had lots and lots of these epileptiform discharges almost in every second. They're in the right posterior quadrant of his EEG. And when And he had some developmental problems and had had a birth injury with an infection. And, you know, when we took a picture of his brain, he had a lesion there. So it's a well-defined biologic characteristic. Here are these spikes. It indicates a pathologic process. And we are hoping this will change with therapy. He's scheduled for surgery in the next few weeks. And so our idea is if we take this out, it will clean this up. And we're hoping that some of his the seizures will get better and potentially some of his behavior will improve as well. I think this slide crystallized biomarker development for me in a way that I'd been struggling with for years, because we talk about biomarkers all the time, and then the conversation is always like, oh, I found this, and oh, I found this, but where does that live? How good is it? Can I use it? Is it ready to go? The NINDS is interested in biomarkers and put this slide on their website to sort of lay out what the process looks like. The first step is discovery. I feel like discovery is sometimes a bad word in our field because people say, oh, it's a fishing expedition. It's not. Give us a break. Discovery is important. It's critical to what we do. Sorry, my pager is going off. It's critical to what we do. In order to find biomarkers, you have to look, and often this requires these broad investigations looking at lots and lots of different candidates to figure out what's going to work. So discovery is important science. Once you have your idea, you need to prove that there's a there, and so there's this sort of analytic validation work that gets done to define what it is that you're measuring. Once you've done that, you need to do clinical validation to prove it in larger networks, larger groups of patients. And then if you want your biomarker to be part of a device or a test or something that you can order and you want to have regulatory approval, then there's this qualification step that goes on where the FDA is going to review it. Okay. That was the foundation. Let's talk now about some data. I want to share with you some natural history work that we did in New York. Laura Deering was a medical student, and she did most of the labor of the work that I'm going to share with you. We got some CDC funding several years ago to gather all of the text of physician notes for children with epilepsy from 5 of the centers in New York: Cornell, Columbia, Montefiore, NYU, and Mount Sinai. What Laura did, she's now Dr. Deering. What Dr. Deering did is she took all of the kids who in the last year of our data, 2014, had a diagnosis of Lennox-Gastaut syndrome and were either 4, 5, or 6. That allowed us to go back and understand everything that happened to those kids from when they were born. We had 5 years of data. So she found 75 cases, 9, and we did a text search to find these, looking for words like Lennox-Gastaut and slow spike and wave and paroxysmal fast, et cetera. 9 of those kids, when we dug into the chart, did not have Lennox-Gastaut after all, leaving us with 66. The charts didn't have a lot of information for 26 of those, and that's a danger with electronic health record data. And so there was 26 charts we just couldn't do anything with. There were 40 left, and then 7 we couldn't really get a good handle on their seizure frequency, so we didn't want to include them either. So we ended up with 33, but with these 33, we had really high quality, multiple notes, sometimes at multiple centers, narrative text about what was going on with them. This is a little information about them, about half male, a variety of causes. 2/3 of this group had a history of spasms, and 1/4 of them had their first seizure in the first 3 months of life. Interestingly, 2/3 of them had a gap in their seizures. Most children with Lennox-Gastaut syndrome in early life often have relentlessly refractory seizures, but and Dr. Berg has written about this, several others have written about this. Many children have this gap. They have really bad seizures in the first year or 2 of life, and then you kind of get them under control, and like they're on 2 or 3 drugs and maybe the diet, and you're kind of good and things are okay. And then suddenly 4, 5, 6, they sort of descend into refractoriness again, and the seizures get much worse. And I think it will resonate with many of the clinicians, that narrative, and with many parents on the meeting, that may resonate with you as well. So what we wanted to do was understand when some key events happened, including that gap. This is an example of one of the graphs we put together. It's a cumulative frequency graph. So on the x-axis here is the age of the child, 0, and then we had some up to age 6. And then the y-axis is really percent. This is like 0% up to 100%. And these lines show how many of the group of 33 have had that thing happen by the time they get to a certain age. So this first green line is when was the first seizure. Half of them have their first seizure by 5 months. And then in this group, all of them had their first seizure before they were 4. This is slow spike and wave. We didn't have the EEG, so we're trusting our colleagues around the city. This is somebody wrote slow spike and wave in an EEG report or in a note. This is the first tonic seizure. Before I did this, I used to say like, oh, if you don't have tonic seizures, you don't have Lennox-Gastaut syndrome. So that's wrong. More than half of these children, almost 3/4, do have tonic seizures by age 6, but not all. And then the status epilepticus is always in our mind, but only a minority of children in this cohort, again, had at least one episode of status epilepticus by age 6. So let's look at that first slow spike and wave. This is one of the most exciting graphs I've seen in my work. And I don't know if I can totally share that enthusiasm with you, but I will do my best. Remember how I told you about that gap? So this is the cumulative incidence plot of when the gap begins in this cohort. Of the 22 kids that had it, it's about 2/3 of the kids. This is when their gap began, and this is when the gap ends. So this is a period of time between 6 months and maybe 4, where these are children who had seizures, who are no longer having seizures, but Lennox-Gastaut and relentless refractoriness is in their future. The gap begins, the gap ends, and right in the middle of it is the first slow spike and wave. So what this means when I saw this is that there's a missed opportunity. There are children who have really ugly EEGs, and ugly, I mean what Dr. Patel talked about, and there was another speaker who talked about, it was in reference to Kelly Knupp, where you're looking at the EEG and you've got that feeling in your gut like, oh, this kid is headed towards Lennox-Gastaut because it's kind of headed in that direction. In our cohort, some of the epilepsy docs in New York City saw that and began to call it slow spike and wave. So this points to an opportunity. We could find these kids, the high-risk ones, and we could just monitor, do some serial EEGs, and when they have that slow spike and wave, do something, even if they're seizure-free. The unanswered questions for this concept is, what is high risk? What do we mean when we say slow spike and wave? And are parents going to be interested in us doing some very aggressive thing, high-dose steroids, starting vigabatrin, epilepsy surgery, devices for their child who is not having seizures? I don't know the answer. After I found this, I had a patient and I started pitching the family about how we were going to do this high-dose steroid regimen, and they never made a follow-up with me. So I don't know the answer, and I want to take a little bit of a detour for this second question, inter-rater reliability. Dr. Bevan mentioned this, and some other people mentioned this. Inter-rater reliability is not obvious. We keep talking about slow spike and wave like we all know what that is. We don't. Hypsarrhythmia is our cautionary tale. As a medical student and as a resident, all these fancy doctors who I still look up to are like, oh, this is hypsarrhythmia. Oh, this is modified hypsarrhythmia. And I'd kind of squint my eyes and I'd be like, it looks bad, but how do you know? Here's an example. This is hypsarrhythmia, right? We all know. Look, it's got multifocal spikes, and when you zoom in, it's got high amplitude slow waves. So multifocal spikes and high amplitude slow waves, that's clearly hypsarrhythmia. My friend, my colleague, Sean Hussein, showed that this was misguided. He took 5-minute snippets of EEGs, and he sent them to 6 different epilepsy docs, many of whom are some of the leaders in our field. And he said, is this HIPS? Is this hypsarrhythmia? And the agreement was not that good. Is it abnormal? Okay, sure. The kappa scores were pretty high. But was it hypsarrhythmia? Well, in his centers, the agreement was like medium, 0.5. That's not awesome. But among the non-UCLA raters, so not at his center, 0.22, so not great. And then when you took the 6, the inter-rater reliability is 0.4. Dr. Bebin, in her, when I've spoken with her, has talked about inter-rater reliability for the PREVENT trial for epileptic spikes, not hypsarrhythmia, epileptic spikes, to be 0.8, 0.85, 0.9 in that range. And other people have said similarly. But this pattern, hypsarrhythmia, was not that good. This is a solvable problem. This is not the end of the world. John Mightinger at Nationwide Children's Hospital figured out how to do this. He came up with something called the BASID score. And in their first go, they found that readers using his score, using his system, were able to have agreement with a kappa of 0.87. That's a 93% agreement. He has since refined the score as he's used it over the past few years. He's found some ways to tweak it, and they just published that in 2021. So here is his score. For the Lennox-Gastaut world, the lesson here is that slow spike and wave, we need to know what we mean by that. And that's itself a study and a research goal for this community. This was a cute study. I don't mean it in that way. It's in JAMA Neurology. This is an amazing study by the Mass General Group, Brandon Westover, senior author here, where they took epileptiform discharges and they looked to see inter-rater reliability. And I'm putting this up just to show that this is very. There's lots of gradations here. He had 8 reviewers looking at these interictal epileptiform discharges, and then he shows some samples in their JAMA Neurology paper from the past year or two, showing which kinds of squiggly lines on the EEG were called by the 8 reviewers as epileptic or not. And you can see at the top 0 out of 8, and then as you go down, the number increases until at the bottom. And for the epileptologist, some of these are like, ooh, that squiggle, 5 people thought that was epileptic. That looks pretty lame. But what about this one? Ooh, that's pretty sharp. Maybe that should have been called by more people. So figuring this out, figuring out how to measure this is going to be really important. These are some snapshots from the literature. This is shown as an example of slow spike and wave, and so is this. This is my patient that I showed before. So I didn't call this slow spike and wave. I called this ESES, epileptographic, the electrical status epilepticus of sleep, or continuous spike wave in sleep. But if I showed this to a room of 10 epileptologists, how many of us would call this slow spike and wave? I don't know. Okay, so that's opportunity 1, is really focus on that slow spike and wave. 2 more opportunities to think about. A quarter of the kids have their first seizure in the first 3 months of life. Dr. Bebin has a study that's here in the US looking at using vigabatrin to prevent seizures in tuberous sclerosis. I'll briefly just highlight the EPISTOP trial, which did the same. They enrolled kids with tuberous sclerosis under 4 who had not had seizures, did monthly EEGs, and then if the epileptiform discharges appeared, they were randomized. Well, they were either given vigabatrin or just followed clinically in case they had seizures. They had a variety of outcomes. There were some wrinkles with that study, only 6 randomized, and then the other 2 sort of chose one path or another. And they were able to get 94 in the study, 54 with those epileptiform discharges. Their outcomes are remarkable, which is why I wanted to show them. You know, when you look at cancer trials, you see these Kaplan-Meier curves sort of gently spread. But in this case, there's this really dramatic change where the percentage of patients who were seizure-free in the preventative group by the end of the trial was 46%, versus 15% in the ones who were just watched clinically. And the dramatic nature, that kind of effect size was also seen with drug-resistant epilepsy, 28% with preventative versus 64% conservative management. Infantile spasms, none in the preventative versus 40% in the conservative. And then the other outcomes, developmental delay and autism, the direction of effect favored VIGAP, but it wasn't statistically significant. And then they don't report a lot of adverse events. And this is just to give Dr. Beban a shout out for her really remarkable trial where she's working on a similar question to flesh this result out. And I am personally eager to hear what we learned from that trial. We have done similar work in using the neonatal seizure registry. Dr. Peters gave us a shout out that I was grateful for. Hannah Glass at UCSF and Renee Schellhaus at Michigan Medicine got a cohort of 204 neonates with acute neonatal seizures with a variety of etiologies. I worked with the 2 of them and their group and a PhD statistician to develop a risk prediction model. And we used a lot of fun algorithms. We used the best subsets method. We used some bootstrap analysis to develop a risk prediction model in this group, a broader group of children with acute symptomatic seizures, not just tuberous sclerosis. Actually, I don't think we had TS. In this group, to figure out which of these kids would get spasms by 12 months of age. 12 of those kids, 6%, did develop spasms, and we were able to come up with a way to identify them. A little bit complicated. Did they have a severely abnormal EEG? And we had a definition for that. Or did they have EEG seizures on 3 or more days in the neonatal unit? That's one criteria. Did the MRI show deep gray or brainstem injury? That's the second. And then third, clinically, when they discharged and you did the exam, was their tone abnormal? If they had all 3 of these, then more than half of those, 8 out of 14, developed infantile spasms. So you remember that chart with the biomarker list? We did the discovery. We looked at a bunch of different things and we came up with an idea. This needs validation, both analytic validation and clinical validation before it's really ready for prime time. But it does prove the principle that using clinical measures, we can identify in a large group of at-risk kids who's likely to develop infantile spasms. And then the third opportunity, and I know I'm running short on time, is that a quarter of the kids developed their first seizure between 3 months and 5 months, which is really like prime infantile spasms time. So there are a ton of questions with infantile spasms that are unanswered. This is just a time frame of infantile spasms. You have babies who are born, they become an infant, and then sometime during their infancy, they have infantile spasms. We know really well what that first line treatment is, ACTH, prednisolone, or vigabatrin, alone or in combination. If the spasms continue, we don't really know what the second line treatment is supposed to be. A little bit of data suggests that you kind of use some mixed mechanisms, but that's not been yet confirmed. Sometimes the spasms remit and you're like, woohoo, the spasms are gone. But then a percentage of those kids will relapse and have spasms again. So all throughout this time frame, there are questions that need answering. Which neonates rather, are going to develop infantile spasms? Can we confirm that we know that? When they're infants, are we doing things to increase the risk of infantile spasms? Sean Hussein again published a paper saying like, ooh, maybe when we use sodium channel blockers, that's going to increase the risk for infantile spasms. We need to confirm that before we change our practice. But that would be an easy way to change our practice and then potentially avert some cases. Who responds to therapy? What's the best second line therapy? Can we prevent relapse? Can we predict relapse? So let me summarize with my last 30 seconds. To summarize what I've said, there's some opportunities for research with existing data sets. The health data infrastructure itself needs to be strengthened, and there's lots of work to be done to make sure that in our routine care, we're putting in data that's usable. There are lots of large data sets available for comparative effectiveness, natural history, et cetera. And then all those biomarkers need work. For prevention, there are these three opportunities. Aggressive treatment of slow spike and wave, even if seizure-free, early identification of high-risk neonates, and optimizing care for infantile spasms. Thank you all for the chance to speak with you today.